Hello everyone. Welcome to our Translation Neuroimaging educational program. I'm the moderator for this event today, and my name is Riyang Ge, and I'm currently working at the University of British Columbia, and I will be joining the University of Macau in a few months. And before introducing our guest speaker for today, please allow me like 2 minutes to have a quick introduction of this educational program. So this education program is nested within the lifespan working group of the Enigma Consortium, which spans more than 45 countries and involve over 4,000 researchers worldwide. This is a very inclusive program that welcomes researchers at all levels of neuroimaging skills and from diverse training background. So the director of this program, Dr. Shalala Haas from Mount Sinai in New York City, and myself, and we do have an international advisory committee. Please consider joining our mailing list if you wish to receive notification of updates about this program. And in addition to our online workshops like today, we also organize in-person workshops. For example, over the past 2 years, we have already hosted free in-person workshops at the SOBP annual meetings. We have a YouTube channel that you can find the recordings from previous event. I'm also recording today's event, so if you are interested, keep an eye on our YouTube channel. And just a heads up, we will only be sharing the presentation by our guest speaker, not the QA part. So please feel free to raise any questions for our guest speaker. We are also both on X and Blue Sky. And if you are on either platform, we would love you to follow us there. If you wish to share your work through this platform, please get in touch with us. We're happy to showcase and highlight your high-impact research work. Our next event will be August 31st, when Cal from University of Melbourne will join us to share their latest work on white matter pathways for TMS therapy for depression. And today, we're very excited to welcome Jen Ye. Jen is a neuroscience PhD graduate student at Yale, working with Dr. Dustin Shinoist and Elizabeth Goldfarb. Her research examines how moment-to-moment brain responses support flexible behaviors. And Jen is really a rising star in this research field, and she has published as the first author in many high-impact journals like Neuro, JAMA Network Open, and Biological Psychology. So in this talk, Jen will introduce the state engagement variability, SEV, which is a normal measure of neural variability that captures fluctuations in the recruitment of the recurring brain states. She will present evidence linking SEV to typical development, executive function, and mental health. So Jen, we are really looking forward to learning about your work and your insights. And the microphone is now yours.
Well, thank you so much for the introduction. So I'm going to go ahead and share my screen now, and also really excited to be here to share some of the work that we have done as mentioned earlier. Hopefully you can see my slide, or actually I don't know if you can. Okay.
Not yet.
Okay, 1 second. It seemed to have stopped sharing for a second. Excuse me. Ooh. Okay. Let me do that again. Okay, so hopefully you can see it now.
Yeah.
Okay, great. So as I was saying before, thank you so much for the very kind introductions. And we're really excited to be here today to share some work we have done looking at neurovariability in the context of development and also in psychopathology. So throughout the day, we encounter a really rich influx of information, and we often have to decide on how we're going to respond to these different information. For example, you might be at home cooking, and as you're cooking, you hear your cat in the other room, and you most likely guess that it's unlikely to be something really urgent. So instead of paying attention to your cat, you focus on cooking your soup. However, your response may be very different if instead of hearing your cat, you hear this really loud alarm sounding in the room. In that case, you might actually want to stop cooking and pay attention to the alarm instead. And as you can see from these cartoon examples, your response to these information could very much differ depending on what it is. And in some situations, it may be better for you to ignore distractions. Sometimes it may be better to switch tasks. And this behavioral flexibility is often supported by executive function. Executive function is quite crucial for our daily life. It's also one of those behaviors that seem to change quite drastically during development. And this might be beneficial given that adolescence is often categorized by rapid changes in the environment. So improvement in executive function can potentially support an individual in navigating through an increasingly complex world. And there are also a lot of work being done to understand what are the potential neuromechanisms supporting executive function improvement during this time. And one of the possibility is that this behavioral flexibility is also supported by corresponding dynamic moment-to-moment fluctuations in brain responses. And there are also a number of ways to look at brain dynamics. One of them involved looking at neurovariability, which describe variation in brain signals. And the idea here is that greater neurovariability could potentially confer greater flexibility in recruiting different brain network configurations before adopting the most optimal one for the task at hand. And in addition to neurovariability, there are also work looking at brain states dynamic. So after identifying these recurring brain activation patterns, you could potentially look at how individuals recruit these brain states and transition between these brain states. And these are activation patterns that seem to come up again and again when people are doing different tasks. So one thing that we have been thinking about is how we can potentially combine information from both of these domains to essentially look at how variable individuals are at engaging these recurring brain states. And the idea here is that if we can measure this variability, greater variability could potentially reflect greater flexibility in exploring and also combining these whole brain activation patterns in order to effectively respond to evolving demands. So this is the background for today's talk, and I will largely break today's talk into three parts. The first one will be me telling you about a new brain dynamic measure we developed that combine information from both neurovariability and also brain state dynamic, and also to study whether it may be behaviorally relevant during development. So there are two questions that we are interested in asking here. The first one being that we want to see if SEV or this new brain dynamic measure might follow a consistent developmental trajectory. And the second one is if it does, then we can start thinking about whether deviations from this developmental trajectory may have behavioral implications. And for the second part of this talk, I will move on to discussing some newer work that we have done to kind of zoom out a bit to start to think about this brain dynamic measure across the lifespan. And I'll end today's talk with a quick demo to show you some scripts that we have made publicly available in case you're interested in running any of the analysis that we're going to talk about today. To look at variability in brain state engagement, the first thing that we'll need to be able to do is to track each brain state continuously. And that might sound really intuitive. However, it may be difficult to do if each time point is only associated with the most dominant brain states. And this is the approach that many previous methods often take. So to address that limitation, we introduce a framework that allows both temporal and also spatial overlap in brain state engagement. And what this framework will do is that it will take in a set of data set that you're interested in looking at, as well as a set of brain states that you're interested in looking at. And I have four here as an example. And the framework will pull out one time point at a time and use regression analysis to regress the brain states that you're interested in from your time point of interest. And what this step will give you is a beta coefficient, each indicating how much an individual might be engaging a particular brain state at this particular time point. And if you can imagine doing this for every single time point in your data set, what you will end up having for each brain state you're looking at is a continuous assessment of how much an individual might be engaging a brain state over time. And since we're interested in looking at variability, you can then take the standard deviations of that beta coefficients across time to look at state engagement variability. And state engagement variability, or SEV for short, is going to be our main way to look at dynamic engagement of brain activation pattern. And the interpretation for SEV is quite similar to how people have been thinking about neurovariability. So in this case, higher SEV may reflect greater flexibility at the neural level to potentially support better executive function. And as I mentioned earlier, you can use any brain states you want. Here, we decided to replicate our prior work to identify 4 recurring brain states in a human connectome project dataset. And we chose to do this for 2 main reasons. The first one being that, as you can see here, the HCP dataset included a range of tasks, so really giving us the opportunity to identify brain states underlying different cognitive processes. And the other reason being that once we identify these brain states in HCP, we can take them and apply them to other data sets without worrying too much about circular analysis. And I'm not going to go into too much detail into how we identify these brain state. If you're interested in learning more about the methods, I will point you to this particular reference here. But I want to at least show you what the brain state looks like so you know what we're actually working with. Here we use linear manifold learning to first reduce dimensionality of these fMRI data and project them into a lower dimensional space. And here I have a 2D representations of what that lower dimensional space looks like. And each dot here represents a time point, and they're also color-coded based on what task condition they're associated with. And time points showing similar brain response pattern are located closer together. And based on that characteristic, we identify 4 recurring but distinct brain states that seem to come up again and again when people are performing different tasks. And we also label them based on what participant might be doing during those time points. So we have fixation, where participants were looking at a crosshair in the middle of the screen, high cognition, where people were performing relatively high-level cognitive tasks, such as two-back working memory, relation, and social or gambling tasks. We have low cognition, where participants were just moving different parts of their body. And then lastly, we have transition, where participants transition from one task condition to another. And we just use these 4 brain state to extract SEV. And one thing I do want to mention here is that we did validate our results using brain states identified in clinical and developmental populations, and we were able to get pretty consistent results. So for today's presentation, I will focus on presenting results that we have obtained using SEV extracted from the HCP brain states. Okay, so now that I've told you about the brain dynamic measure that we will be using, I'm going to move on to telling you about work that we have done to see if SEV may be behaviorally relevant during development. And the first question that we want to ask here is test the relationship between age and SEV and how it might be changing during development. And the hypothesis here is that if neurovariability supports executive function, and if executive function improves during development, then one possibility is that SEV may also increase with age during this time. And to test this question, we extracted SEV from 2 cross-sectional data sets, the PNC and HBN dataset, both during rest and also during a naturalistic paradigm. And then after we extracted SEV from these data sets, we looked into the relationship between age and SEV. And what we saw in these dataset is that there is a positive relationship between age and SEV. And we saw this for all 4 brain states we looked at, and also in both of the cohorts, both when participants were resting or when participants were watching a clip from Despicable Me. So in both of these cohorts, you're seeing that across these participants, SEV seem to increase with age. Even though you're seeing a lot of positive relationship here, one thing that I would point you to is that the effect sizes or the relationship between age and SEV seem to be larger for the fixation and also high cognition brain state. And these are brain state that seem to recruit more of these heteromodal brain regions in the association cortex. So this positive relationship is really exciting to see and is also aligned with our hypothesis. However, cross-sectional data sets may not be sensitive to subtle developmental changes. So this is something that we want to validate further by next turning to 2 longitudinal data sets. And there's also additionally a questions that we want to ask here, which is that even though we saw the positive relationship on the previous slide, we don't really know if SCV continued to increase indefinitely during development. And this is because executive function itself also doesn't continue to increase indefinitely. People do tend to reach adult level performance once they reach mid to late adolescence. So for that reason, it's also possible that SCV may not increase increase indefinitely during development. And this is the questions that we want to test out further in longitudinal data sets. So the first data set we turn to is the Imogen data set. And for this particular study, participants came in at 3 different time points. So at age 14, 19, and 23 years old. So I do want to mention that this age range is slightly older than the age range covered by PNC and also HBN, giving us the opportunity to see what happens after that age range. And what we saw in Imogen is that for the fixation brain state in particular, SEV was the lowest at age 14 compared to age 19 and age 23. And we didn't see this result for any other brain state, suggesting that the age differences could be different depending on which brain state you might be looking at. And the other observations that we can also make here is that when you compare the 2 older age group with each other, so age 19 and 23, you didn't see a significant difference in SEV. So that suggests that SEV might not continue to increase indefinitely during development. So building upon the results that I showed you on the previous slide, we're seeing that SEV may increase during early development, but might start to stabilize in mid to late adolescence. So this is the result we're seeing in Imogen, which kind of build and give us more information compared to what we saw in PNC and HBN. So the next thing we want to do is to validate the trajectory we saw in the previous 3 data sets in the Michigan longitudinal data set. And the MLS data set is great for validation since for this particular data set, participants were between 7 to 28 years old, so really covering the age range that PNC, HBN, and also Imogen covered before. So it will be really nice to see if we can replicate the same trajectory in this independent data set. And the other thing about MLS is that each participant completed multiple visits, and this scan sampling give us the opportunity to look into smoother developmental trajectory. And once we look into the relationship between age and SEV in MLS, here are the trajectory that we saw. And we were able to replicate trajectory that we saw in the previous data set. Specifically, we're seeing that SEVs seem to increase with age pretty drastically during early development before a peak and a stabilization start to occur in mid to late adolescence. And in addition to the replication, there are also 2 observations that you can make here. The first one is quite similar to the pattern that we saw in the Imogen data set, where we are seeing that the stabilization or the peak could differ depending on which brain state that you're looking at. So in this particular example, we're seeing that stabilization seemed to occur the earliest for the low cognition brain state. And this is the brain state that recruit more of these unimodal brain regions and the sensory motor system. And the other observations that you could also make here is that female participant, which is indicated in the yellow color, seem to reach stabilization earlier than male participant, which is indicated by the green color. And this is something that we can formally test using permutation testing, which reveal that female participants arrive at the peak age earlier for the fixation brain state in particular. So to recap the trajectories that I've shown you so far in 2 different cross-sectional and 2 longitudinal developmental data sets, we were able to identify a pretty robust developmental trajectory for SEV during resting state, naturalistic, and also task-based fMRI data. And the fact that we were able to see this trajectory across these different paradigms suggests that it may be robust to brain response differences induced by different tasks participants might be doing in the scanner. And this trajectory is also categorized by a really rapid increase during early adolescence before a stabilization occur in mid to late adolescence. And there are also a couple characteristics that I also want to mention here. The first one being that the stabilization timing could vary by brain state. So the low cognition brain state, which is the one that engaged unimodal brain regions in the sensory motor system, tends to reach stabilization the earliest, whereas the fixation brain state, which is the brain state that recruited more of these heteromodal brain regions in the association cortex, reached stabilization the latest. And these 2 results together suggest that these result and the patterns that we're seeing SEV very much aligned with the unimodal to heteromodal developmental hierarchy that people have talked about before in prior literature. And the other characteristic that I also want to mention is that female participants seem to reach SEV stabilization before male participant. And this is again a pattern that other people have also seen before by looking at brain features extracted during structural or functional MRI data. And perhaps more excitingly for us, and also kind of linking back to where we started this talk, the trajectory that we're seeing for SUV very much aligned with how executive function changes during development, where you are seeing this really rapid increase and improvement in executive function during early development before people start to reach adult level performance in mid to late adolescent. So really raising this possibility that SUV might be changing during development to support improvement in executive function. However, we didn't really quite look at, or so far, none of the results I've showed you didn't directly look at the relationship between executive function and SUV, which is something that we aim to do in this next analysis I'm going to tell you about. And the questions that we are really interested in asking here is that in the previous slide, I showed you that SUVs show a pretty consistent developmental trajectory. Then one potential questions that we have here is that deviations from this typical developmental trajectory may be related to differences in executive function. And this is the questions that we explore by computing developmental deviations using a brain age approach. Specifically, we are using machine learning models, and here we're just using linear regression to predict brain age using an individual's SEV profile. And we focus specifically on the two cross-sectional data sets, so PNC and HBN, given that the two data sets have pretty similar age range, and having two data sets also allowed us to do external validation. And after we compute this developmental deviation measure, we then computed the square difference between brain age and biological age. And the interpretation here is that a greater developmental deviation is an indication that someone might look younger or older based on their SEV profile. And after we computed this, we then correlated deviation with executive function while controlling for biological age. And what we saw in both PNC and HBN is that there is a negative relationship between executive function on the y-axis and then also alteration in development on the x-axis. One thing that I do want to mention here is that we did regress out age from both executive function and developmental deviation to match the partial correlation that we did. And this is why you're seeing some negative values for executive function. But in both of these cross-sectional data sets, what we're seeing is that greater deviations in SUV development were linked to worse executive function. And this result is pretty exciting for us in that it not only shows us that SUV is quite relevant for executive function, but that SUV is on pace development, so not growing up too fast or too slow based on your SUV profile, is also quite behaviorally relevant as well. Okay, so to recap the first part of this talk, I told you about a new brain dynamic measure that we developed, combining information from both neurovariability and brain state dynamic, and show you that this new measure, SUV, show a pretty consistent developmental trajectory in cross-sectional and longitudinal populations. And I also showed you some work to demonstrate that SUV is quite behaviorally relevant during this time. Specifically, developmental deviations in SUV were linked to worse executive function. So I'm now going to move on to the second part of this talk, which is going to be about some newer work that we have done to kind of move beyond development and start thinking about SUV across the lifespan. And if we can identify a relatively consistent trajectory for SUV, can we use a normative modeling approach to study the behavioral implications of its deviation? But before I move on to telling you about some work that we have done across the lifespan, I do want to take you on a detour and start talking or telling you about some work that we have done looking at SEV and psychopathology. And this is going to make sense in a little bit. And we thought this is interesting to look at, given that executive function alterations are often commonly observed in psychopathology. So one possibility is that we may also see altered SEV in psychopathology. And this is something that we started to look into in a couple clinical populations. And what we saw in these prior study is that individuals with clinical disorder, including those with schizophrenia and bipolar disorder, they did show lower SCV compared to healthy control. And we also saw this during resting state and also task-based fMRI data. The other thing that we also saw on top of this group comparison differences is that in transdiagnostic population, lower SUV was also linked towards executive function. So altogether, these results also give us another line of evidence to show that SUV is indeed quite relevant for executive function, this time in the context of psychopathology. However, the more relevant or interesting part of these results that it's probably more relevant for today's talk, is that when we control for age in our especially group comparison analysis, what we saw is that age had a significant effect on SUV in adult participants. And when we actually plot out what that looks like in these population, we actually saw a negative relationship between age and SUV. And we saw this pretty consistently across different populations. So in healthy controls and also in individuals with clinical disorders, and also during different paradigms. So here we have result from resting state and also a task-based fMRI. So as you can see here, these are quite different from what I've showed you in the first part of this presentation, where we see a positive relationship between age and SUV. And these results really got us thinking about how we can potentially go beyond development and start to think about what happened after that stabilization occurred in mid to late adolescent. We don't really know if things kind of continue to stay high and plateau after a while, or things start to decrease. And this is a question that we set out to test in this next study that I'm going to tell you. For this study, we decided to look at how SUV might be changing across the lifespan. And by lifespan, we just included individuals between 5 to 80 years old from the data sets that I've listed here. The other thing that we also try to do here is to include healthy individuals and also clinical populations to capture as large of a range as we can when it comes to SUV in the population. And this time we collaborated with a lab mate, Chris Kim, using generalized additive model to generate SUV growth curves using information from these data set. So we have data from over 4,000 participants. And here are what the growth curves looks like. And the difference, just to kind of orient you a bit, each of the blue dots here represent a participant, and the rainbow curve represent different percentiles. And one thing that you can already see here is that we were able to replicate previous SCV developmental trajectory, even though the MLS dataset was not included in these growth curves. So suggesting that we were able to identify similar trajectory by pulling participants from all of these cross-sectional data sets. You are still seeing that rapid increase in SUV during early development before a stabilization occur in mid to late adolescent. And this time we also have new information on how SUV might change after that. So we kind of see things start to slowly drop down a little bit. And the pace of that is definitely much less rapid compared to the pace you see during early development. And the other thing that you can do here, now that we have these growth curves, is to look at each participant relative to other individuals in their age group. So for example, if a participant ends up on this green curve here, that means they're on the 50th percentile and have SUV around the mean of the individuals in their age group. If they end up on this purple curve here, that means they're on the 98th percentile and have SCV much higher than most of the individuals in their age group. And just to give you one more example, if they end up on this brown curve here, that means they're on the 2nd percentile and have SCV much lower than most of the individuals in their age group. And having these relatively consistent trajectory across different participants with different ages. And also these growth curves allowed us to start thinking about how we can potentially study the behavioral relevance of SEV deviations using a normative modeling approach. And there are many different ways to do normative modeling analysis. Here, I'm just showing you one example that we have been thinking about using this cartoon right here. And what we plan on doing is that we can first pull participants from multiple data sets to generate SEV growth curves, which are the ones that I showed you on the previous slide. And what you can do next is to project participants from an independent data set onto these growth curves to look at deviations. And this is quite similar to when you were younger and go to a doctor's appointment and they take your measurements to compare with people in your age range. And lastly, after you extracted these deviation measure, you can correlate them with behaviors of interest. And this opens up to many different questions you can ask depending on what behaviors you might be interested in looking at. And I'm going to show you one particular example, and this is very much ongoing work, looking at combining our interest in development and psychopathology to look at whether SEV deviations may relate to risk factors for psychopathology and clinical symptoms during development. And to look at this question, we look at individuals with depression and anxiety disorder from the BANDA dataset. So we follow the pipeline that I showed you earlier. We used the growth curves that I showed you in the previous slide, and we projected BANDA participant onto these growth curves to look at deviations for each BANDA participant before relating that deviations with risk factor score and also clinical symptoms. And before I show you any result, I just want to orient you to the axis. On the x-axis, we have behavior. So we are looking at family risk as one particular form of risk factor. And then also internalizing symptoms as our clinical symptoms of interest, given that these are individuals with depression and anxiety disorder. And then on the y-axis, we have SEV relative to peers. And first, looking at risk factor, we are seeing that there is a positive relationship here. So individuals with higher family risk seem to show higher SEV relative to their peers. And the potential interpretation here is that when you are in an uncertain environment, it might be beneficial for you to become more flexible in order to navigate this uncertain environment. And the other thing that we also saw is that having greater SEV than peers could potentially have implications for clinical symptoms in a sense that individuals with greater SEV seem to show fewer internalizing symptoms. So being flexible and being able to navigate an uncertain environment could potentially be beneficial for others to regulate, for yourself to regulate your emotions. But this is one example that we have done, and as I mentioned before, still very much ongoing work, and we're interested in also thinking about whether deviations in SEV may relate to executive function. And this could be potentially interesting to ask, given that executive function also continued to change after people move into adulthood. So we're interested in looking at whether SEV deviations may relate to executive function differences in adult population and also as people move into aging populations. Okay. So this is the second part of the talk, and just to kind of wrap things up a little bit, because I think this is all the data I'm going to show you before I move on to the demo portion. So here we are showing you that SEVs seem to change in consistent way across the lifespan in multiple cross-sectional data sets, and having these relatively consistent trajectory give us the opportunity to then use a normative modeling approach to study behavioral implications of SEV deviations. So now I'm going to move on to showing you some codes that we have made publicly available in case if you're interested in extracting SEV from your own
Sorry,
data
Jen.
set or try doing the auto. Yeah, yes.
Just lost your voice for a few seconds. That's
Oh,
fine
okay.
now. Thank you.
Okay, that's great. I was just saying that I'm gonna show you the script that we have made available on GitHub. And first to show you some script that we have uploaded to extract SEV, and then also some script showing you how you can look at deviations in SEV. Okay, so all the script that I'm going to show you are available on GitHub, and most of the script that I uploaded on there are written in MATLAB, but there are some written in R as well. And the first thing that I will point you to on this under this kind of link is this functions that we have that you can use to extract SUV. And there is some descriptions that we have made available telling you what input this function takes in and also what output it gives out. And just to briefly go over it a little bit, for this SEV function, it would take in two different inputs. The first one is the time series matrix from each participant, and each row here indicates a brain region and each column indicates a time point. And then you also want to give this function a set of brain states that you're interested in looking at. And you can give it any brain state you want to look at depending on what your research question is. But we have also uploaded the brain states that we have been using from the HCP dataset to GitHub under this particular path. And the brain state was parcellated using the SHIN260 atlas. And just to show you a little bit of what might be going on inside the SEV function in case you ever need to go in here to change anything, we first have a for loop where we go through each time point at a time and basically perform the cartoon regression analysis that I've shown you earlier, where we regress out the brain state that you're interested in from the time point. And here we're using non-negative least square regression just because a positive correlation is easier for interpretation purposes here. So we opted to go with a non-negative approach. And after we computed continuous brain state engagement for each of the brain state that you're interested in looking at, there is another for loop inside this function that will actually compute the standard deviations and continuous engagement over time. And that leads me to telling you about the output of this function. The first one is the SV values itself, and you will get one for each brain state you include in your model. And then there are also two additional outputs that I didn't quite talk about today, but they're available to you in case you're interested in looking at them. The first one is looking at how much each brain state was actually engaged. And this is standardized or thresholded by the total amount of engagement over time across the different brain states. And then we also have another output telling you how many time points show an outlier residual, which is something that could be helpful if you want to exclude any specific time points or any participant. So this is just for QC purposes. So the SEV function should be able to help you compute SEV for a participant, but if you're interested in looking at how we can compute SEV from multiple participants, we also have two scripts here available to you. We have one from HBN, which is where we analyze the movie data. So here we didn't actually take out any time points with high motion, just so we can kind of conserve the continuous nature of this paradigm. So we didn't take out any time points. But if you want to take out any time points with high motion, we also have the PNC script here available where we actually did scrub out high motion volume before computing SCV. So we have two examples here in case you're curious. So those are all the script that we have made available when it comes to computing SCV. And we also have some script available to you if you want to look at how we did the deviation analysis. First, the brain age approach. So under this particular directory, we have two different scripts. We have one script where we train the model in PNC before applying it to HBN, and then also another one where we flip the two data sets. Here I'm showing you PNC to HBN as an example, but the structure of it is quite similar for both of them. So the first part is where we train the linear model to predict age using SEV in a training data set, so PNC in this case. Before we take that model and apply it to our testing data set, which is HBN, to predict age in these external participants using SCV from these individuals. And then lastly, we have a section of code here where we actually compute the square difference between predicted age and biological age to look at deviations. So this is the brain age approach when it comes to computing SCV deviations. Next, I'm going to show you some resources we have made available when it comes to the growth curve approach. So Chris Camp, who is the researcher and expert in the lab who did all of the gamma analysis, is building a Shiny app, and this app should be available soon through this link. And for this app, you should have the option to upload a data file and use a drop-down menu to select what output variables you're interested in, and also any additional intermediate output variables you are interested in collecting for any QC purposes. So this should be available hopefully soon, but in case you're interested in looking at the code anyways, we have also made that available on GitHub right now. But for this script, you might need to go in there to do some additional editing before you can use it for your analysis. We just want to make this resource available before the Shiny app is available. And I do want to kind of walk through very quickly what is in the script in case you do want to use it on your own data set. What the script will do is that it will first fit a model using the gammas function in R, and Chris has some really nice descriptions here to tell you what parameters you should edit, and then also a couple family distributions or parameters that you can change up. And there are a couple examples here on how you can run a gammas model using age and any brain measures you might be interested in using different parameters. And if you're interested in looking at what parameter we actually change and what family distribution we actually test out, you can check out a detail list in Chris's paper. And after generating all these different models, we picked the one with the lowest BIC value. So the first session of the code will build that gammas model for you. And once you have a model that you're happy with and that has really that you have validated and gives you good results, there are two additional functions that are available inside the script that can first let you get the C-score or centile score, depending on which one you're interested in looking at from your gammas model. And then if you have more questions about what output this particular function provides you, Chris also has a nice descriptions of the different outputs this function will give you. And another functions that I want to point you to is the plot centile function, which is the function that we use to generate the different rainbow curves that I showed you on the previous slide. And here I also do want to mention that you can also change out any percentiles that you want to include. So if you want to include fewer or more percentiles than what we have here, it should be relatively easy to specify and personalize. So I think that's all the code that I'm going to show you. If you have any questions about anything, feel free to reach out and we'll be happy to answer any questions you have. And with that, I would like to end here and thank you all for listening. I have the codes here again, and then also the papers that I talk about today. And I'm happy to take any questions now. Thank you.